Okay, so today we're going to go over DNA structure, following up from yesterday's lecture on kind of determining that it is in fact nucleic acids or DNA and RNA that are being able to pass on this genetic information from generation to generation. Now that we've discovered that it is in fact nucleic acids or specifically DNA, now the most important part was discovering their structure. Okay. Now, as we go through this, I realize that my slides aren't perfect. My slideshows are still from when we were in regular class, and I haven't had enough time to basically start converting them. So as we go through the course, I will get better at this. I'll also be able to get a little bit better at presenting and going on video. But obviously, as you know, this is kind of my first time through it. But as we progress through the course, I will get better at this. Okay, so I know some people are maybe frustrated when we look through my lectures, being like, well, if we don't need to know this material, why is it in the lecture? Okay, and it's just because I haven't been able to adapt yet to this online scenario. Okay, so I apologize, but I will be getting better. Okay, so let's go through discovering DNA structure. So we finally know now for sure, okay, that it is DNA, and it's not protein. And again, that was thanks to Griffith, Avery, and Hershey and Chase. And I will be giving you kind of some update videos on those specific experiments, because those are kind of the big part from yesterday's lecture to know is how those three experiments work together to prove that it was DNA, not protein. Okay, so we've known about DNA for a long time, but we didn't actually know what it did. Okay, so not too much information on this. Okay, so who discovered the macromolecule? This guy name was Friedrich Michener. Okay, and all you really need to know from this slide is that he is the scientist that's credited discovering the macromolecule DNA. Okay, and he identified it basically from the pus bandages of some of the patients that he was working on. Okay, so Friedrich Michener discovered the macromolecule DNA. Okay. Discovered that the macromolecule nucleic. Okay, now extending that. Once we've discovered the structure of DNA, another scientist named Phoebus Levine discovered that there's actually two types of nucleic acids, DNA and RNA. So the big part of this lecture is just knowing the contributions, okay? I'm not going to ask you to know all the individual details that I would normally do in class, but right now, for this lecture, what we really need to know is what the contributions are. So Michener, going back acids, then Levine discovered that there's actually two types of nucleic acids, DNA and RNA. Okay? And we should already know the differences between DNA and RNA from our previous lectures. Okay? So on the left side, we have DNA here, and then on the right side of the picture, we have RNA. Okay? So we've got Michener discovers macromolecule. Levine discovers there's two types of the macromolecule, RNA and DNA. Okay, so years later, now we're starting to explore this a little bit more. So we know that DNA and RNA are actually made up of this monomer called a nucleotide. So this should be familiar to us from our first unit, okay, of nucleotides. So it's really just a combination of a phosphate. Here, let me just get my highlighter here. So I've got my phosphate here. Then I've got my sugar, and then I've got my nitrogenous base. And those are the three things that contribute to a nucleotide. And this just gets repeated over and over and over again. Okay, so you should know that the two options for the sugar are ribose for RNA and then deoxyribose for DNA. And that comes right in the name, okay, with the R for ribose and then the D for deoxyribose. And then the differences in the bases, those are also important. For DNA, you should know that there's A, G, C, and T. And then for RNA, there is no T, there's only U. Okay. I'm not going to ask you too much about purines and pyrimidines, so don't worry about that. But you should know that A and G have a double-ringed structure. Okay, and what I mean by that is if you look at A and G, right down here, okay, see that they are two rings. Okay, so their bases are composed of two rings, whereas the other ones, C, T, and U, okay, up at the top here are a single ring. So you can never have two single rings combined together. You can never have cytosine and thymine combined together. Okay, you have to have a double ring combined with a single ring. So adenosine combines with thymine. Okay, if you kind of look through those two, you have a double ring bonded to a single ring. And then guanine bonds to cytosine, a double ring and then a single ring. Right? And all the way up, it has to be like that. You can't have a guanine, a double ring, combined with an adenosine, also a double ring. Okay? All right. Now, another scientist. So we had Michener discovers macromolecules. Okay, Levine discovers that there's DNA and RNA. Now our next scientist, Chargaff. And what Chargaff really kind of discovered, okay, to put it simply, is Chargaff really discovered that the bases are proportional to each other. So this is how we discovered that A has to bond with T. Because what Chargaff did was he basically looked at, well, how many A's do you have and how many T's do you have in your body? How many G's and how many C's? And regardless of the organism that he looked at, if you look, A and T numbers are always the same percentages or close to it. Right? And G and C are also close percentages. Right? So this is, Chargaff is the one that basically told us that A must bond with T and G must bond with C. And how he figured that out is because there's equal proportions of each. There's equal proportions of A and T. So that means that every time you have an A, you must have a T. And every time you have a G, you must have a C. So Chargaff is responsible for basically telling us that A must bond with T and G must bond with C. Okay, so now we're getting closer to the structure. Okay, we're closing in on the actual model. So just kind of recapping, we got Hershey and Chase along with Avery and essentially Griffith as well, as the DNA is the hereditary material. Levine, that there's DNA and RNA. Chargaff, now that there's equal amounts of A and T and equal amounts of G and C. So now we're going to start putting all of this information together to try and figure out the actual structure. And this is by James Watson and Francis Crick. Okay, so some other scientists that we want to add to this, okay, is Pauling and Rosalind Franklin. So what did they add in? Right? Well, Pauling was the guy that kind of basically suggested that it's probably a double helix, the DNA. Okay? They didn't really know whether DNA was a triple helix or a single helix. Pauling's contribution by looking at bond angles was basically suggesting to them, not for sure saying, but suggesting to them that the DNA is probably a double helix. Okay? So then Watson and Crick started using this information and saying, okay, well, let's try a double helix. Then we have Rosalind Franklin and Maurice Wilkins. And these two added a couple more pieces of information. So they were using X-ray diffraction, and what they really contributed to the model. So I'm not going to again require you to know all this information in here, but basically, Rosalind Franklin and Maurice Wilkins have now added more information, and what they added was that for sure it is a double helix. So Rosalind Franklin and Maurice Wilkins said, okay, thanks for suggesting it is a double helix. We have now proof it is a double helix. So that's one contribution by Rosalind Franklin and Maurice Wilkins, was the proof that it's a double helix. And then the other huge piece of information was that the bases are on the inside of the double helix, and then the sugar phosphate backbone is on the outside. Okay, so those are the big contributions by the scientists. And then putting this all to Maurice Wilkins. Okay, all of these people help them discover. Watson and Crick eventually put their model together and discovered the structure of DNA. But again, it was only because of all of these other scientists that have helped them along. They got the credit for it, but they were only able to create their structure of DNA based on the work of Levine, of Francis, of this structure. Okay? And this is what we have, basically the final structure. Okay? Now, I'm going to ask you to basically at some point in an evaluation be able to draw DNA. Okay? And when I ask you to draw DNA, it's not going to be to the level of, let me see, it'll be very similar to this. Okay? This is the kind of level that I need you to be able to show me. You need to be able to show me a little bit more detail that there's a sugar phosphate backbone in here, but again, I'll show you the drawing. But then just showing me the bases. I'm not going to ask you to find a picture here, something like this. Okay, I'm not going to ask you to draw to this level. This is way too much detail. Okay, the level that I'm going to be requiring you to draw it, I will draw an actual picture for you to show it. Okay, but we're going to be the actual structure, like to be able to draw these A's and G's and C's and T's. Okay. All right, the other thing I wanted to mention on this is the idea that DNA runs anti-parallel. And what I mean by anti-parallel is there is one strand going one direction. Okay, so let me just highlight. This is one strand of DNA. Okay, and then I have my other strand of DNA on the other side. And what they are is they're anti-parallel, meaning that one is going one direction and one is going the other direction. Now we indicate these directions by calling them 5' and 3'. Well, how do you know which one's 5' and which one's 3'? Well, you look at the sugars. Okay, so when we look at the sugars and we look at the one I've got highlighted in blue here, so let me go back to my blue highlighted. Okay, so on the left-hand side, I know that this is oriented in a 5' to 3' direction. And how I know this is I basically look at my sugars, I find my oxygen, which is right here, and then I start counting to the right. There's 1 carbon, 2nd carbon, 3rd carbon, 4th carbon, 5 carbon. So at the very top, it's 5. And then at the very bottom of the sugar, it's 3. So I've got my 3 right here, and then my 5 right here at the top. So what this is telling me is that 5 is at the top and 3 is at the bottom. So I say this is 5' to 3'. Okay? 5 being at the top, because that's my 5 is at the top, and my 3 is at the bottom, so it's 5' to 3'. Now, on the other side, if we look, and we look at here's my oxygen on my sugar, and I start counting to the right of that oxygen. Carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. Well, now I have my, let me just get it right in here. I have my 3 at the bottom, and my 3 at the top and 5 at my bottom. So now it is 3' to 5'. So this strand right here is 3' running to 5', and this strand right here on the left side is 5' running to 3'. Okay? So I would expect you to be able to label the 5' and 3' on the drawings, but again, I will be showing you the drawing coming up. Elementary. Okay? If we look at the one strand on the left here, we see that the bases are T, G, C, and A, okay, on the left strand. On the right strand of the DNA, it's not exactly the same. Okay? This is T and this is A. They're not exactly the same. They're complementary, meaning that if this is T, that has to be A. If this is G, that has to be C. If this is C, that has to be G. So what you can do is, if you know the one strand, then you can predict what the other strand would be, knowing that they have to bond up like this. T and A have to bond together. There's no way if I have a T here that I'm going to have a G on the other side. Right? So that's what if you look between the AT, let me highlight this again. I'll use a different color. I'll use green this time. Between AT, okay, you've only got a double hydrogen bond, and then between GC, we've got a triple hydrogen bond. So it's a lot harder to break apart a GC than it is to break apart an AT. And we'll come up to that when we do replication. Okay, so the key attributes of the modern DNA molecule. This is basically if we have the structure of the DNA. This is important, okay? I'm going to highlight this because this is basically what our DNA molecule looks like. And I'm not sure why all this other random writing is on here. I apologize. Okay. So let's go through this. DNA is 2 polynucleotide strands forming a double helix. We should know that DNA is a double helix composed of 2 strands. Okay? And they're polynucleotides because they're just poly meaning repeating, and then nucleotide is what is being repeated. Nucleotide is the monomer. Okay? Each strand contains a sugar phosphate backbone with bases connected to the sugar and facing inwards. Okay? The strands are not identical, but rather complementary, called complementary base pairing. Therefore, knowing the sequence of one strand allows you to predict the sequence of the other. I'm going to go back and explain what I mean by complementary base pairing. Hydrogen bonds. Okay, the 2 strands are an anti-parallel, meaning one strand is oriented in one direction and the other one is the opposite direction. That's the whole 5' to 3' thing that I've already mentioned. And that's pretty much it for the DNA strand structure. Okay, so when we look at this, this is a good picture of of what I would expect you to be able to draw. Okay. Now, a couple things I'm going to cross out here that I wouldn't expect you to label, but this picture I would expect you to be able to replicate. Okay, minus a few things. Okay, so I'm going to cross out the things that you would not be able. I would not be expected you to be able to put on there. Okay, let me just make this a little bit thicker. So you wouldn't need to know to label purines and pyrimidines. Okay, you should know about the hydrogen bonds and be able to label hydrogen bonds throughout there. Okay, you wouldn't have to show me the actual phosphate structure. All you would have to put here is just a P. Okay, so just a P. I'm not expecting you to show me the actual structure. Okay. You should be able to draw that sugars in just that shape. Okay, not including the oxygen and the carbons, but just that shape of sugars, because that's what the shape of sugars are. And we should be able to draw those, and making sure that one sugar is upright and one sugar is upside down. You should be able to label the 3' and the 5', right? And then be able to show me that G's are double bases, right? And A's are double bases, and then the T's are just a single base. Okay? So I would expect you to be able to redraw this into this level of detail. Okay? And I know it's can be a little bit daunting just looking at it, but after some practice, this would be the major part of this lecture, is being able to, well, can you draw a DNA molecule? Okay? So again, this is kind of what I'm asking you to do here. Okay, so these are the scientists that I need you to know of what their contributions were, and as we went through it through their lecture. So, I mean, this does a little bit of the lecture 1 on discovering DNA, because you can see that there's Hershey and Chase in here. But these are the scientists that we need to know. Now, the big thing, I know a lot of you are like, geez, there's a lot of material here. This is what I need you to know from our last 2 lessons. 1, I need you to know the contributions of Griffith's experiment, Avery's experiment, and Hershey and Chase's experiment. Okay? You need to be able to explain those 3 experiments. Okay? You then also need to be able to tell me what Pauling contributed to the DNA structure, what Chargaff did, what Watson and Crick, what Michener and Levine did. Okay, so that's 2. So you need to be able to explain to me the 3 experiments, Griffith, Avery, and Hershey and Chase, and then explain to me what these other scientists have done in terms of structure, and then to be able to draw a DNA structure. And those are the 3 things that I need you to be able to do from today's, or the last 2 lessons. Okay, I know there's a lot of other material inside of it, but those are the 3 crutch things. Being able to tell me the 3 experiments, the contributions of these scientists, and then being able to draw a DNA structure. Okay? The rest of this stuff, the rest of this slideshow, I know there's material here, you don't need to know any of the rest of this material. Okay, this is all getting cut because of the situation that we're in. Okay, so none of this stuff right here I'm requiring you to know. Okay, so you can just X this. I've included it just because I don't want to destroy my lectures, because next year when we're actually in class I will teach this stuff, but nothing required here. Okay, thanks guys, and again, let me know if you have any questions.